Hello friends, welcome to Medical Devo. In today's video, we will read the 15th chapter of 11th NCERT, Plant Growth and Development. We will discuss the important point regarding NEET exam. So without further delay, let us discuss the topic. We have already studied the organization of a flowering plant in chapter 5. Have you ever thought about where and how the structure like root, stem, leaves, flower, fruits and seeds arise, and that too in an orderly sequence? You are by now aware of the term seed, seedling, plantlet, mature plant. You have also seen that trees constitute to increase in height or girth over a period of time. However, the leaves, flowers and fruits of the same tree not only have limited dimensions but also appears and falls periodically and sometimes repeatedly. Why does vegetative phase precede flowering in a plant? All plant organs are made up of a variety of tissue. Is there any relationship between the structure of cell, tissue and organ and the function they perform? Can the structure and the function of these be altered? All cells of a plant are descendants of the zygote. The question is then why and how do they have different structural and functional attributes? Let us discuss. Development is the sum of two processes, growth and differentiation. You have to remember this, that development means growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote fertilized egg follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produces root, leaves, branches, flowers, fruits and seeds. And eventually, they die. Figure 15.1 represents this. The first step in the process of plant growth is seed germination. First step is seed germination. The seed germinate when favorable conditions for growth exist in the environment. In the absence of such favorable conditions, the seed do not germinate and goes into a period of suspended growth or rest. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. In this chapter, you shall also study some of the factors which govern and control these developmental processes. These factors are both intrinsic, internal, extrinsic, external to the plant. Here they have represented germination and seedling development in bean. This is the soil line, hypocotyle. Below cotyledon portion is called a hypocotyle, while above portion is epicotyle. Seed coat, epicotyle hook, cotyledons. Epicotyle. Below portion is hypocotyle. Let us see the growth. Growth is regarded as one of the most fundamental and conspicuous characteristics of a living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its part, or even of an individual cell. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occurs at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of a piece of wood when placed in water? This is also one type of growth. Plant growth generally is indeterminate. Plant growth is unique because plants retain the capacity for unlimited growth throughout their life. This ability of the plant is due to the presence of meristematic at certain location in their body. The cells of such meristems have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide, and such cells make up the plant body. This form of growth, where new cells are always being added to the plant body and by the activity of the meristem, is called the open form of growth. What would happen if the meristem ceases to divide? Does this ever happen? If meristem ceases to divide, there is no growth in plant. In chapter 6, you have studied about the root apical meristem and the shoot apical meristem. You know that they are responsible for the primary growth of the plants and principally contribute to the elongation of the plants along their axis. You also know that in dicotyledonous plant and gymnosperm, the lateral meristem, vascular cambium, cork cambium appear later in life. These are the meristem that causes the increase in the girth of the organs in which they are active. This is known as secondary growth of the plant. Figure 15.2 represents the secondary growth in plant, that is increase in girth. Diagrammatic representation of location of root apical meristem, shoot apical meristem and vascular cambium. Arrow exhibit the direction of growth of cells and organs. So the direction of growth is downward in shoot apical meristem, while in root apical meristem it is upward. Here is root, vascular cambium, and upper part is shoot. Growth is measurable. Growth at a cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume, and cell number. You have to remember the parameters which are used to measure the growth, that is fresh weight, dry weight, length, area, volume, cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase in size by up to 350,000 times. In the former, growth is expressed as increase in cell number. The latter expresses growth as increase in the size of the cell. While the growth of a pollen tube is measured in terms of its length, an increase in surface area denotes the growth in dorsolateral leaf. Let us see the phases of growth. The period of growth is generally divided into three phases, namely meristematic, elongation, and maturation are the three phases of growth. Meristematic phase, elongation phase, and maturation phase. Figure 15.3. Detection of zones of elongation by the parallel line technique. Zones ABCD immediately behind the apex have elongated most. Let us understand this by looking at the root tip. The constantly dividing cells both at the root apex and the shoot apex represent the meristem phase of growth. The cells in this region are rich in protoplasm, possess large conspicuous nucleus. Their cell walls are primary in nature, thin and cellulosic with abundant plasmodesmatal connection. The cell proximal, just next away from the tip to the meristematic zone, represent the phase of elongation. Increased vacuolation, cell enlargement, and new cell wall deposition are the characteristics of the cells in this phase. Further away from the apex, that is more proximal to the phase of elongation, lies the portion of the axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening, protoplasmic modification. Most of the tissue and the cell type have studied in chapter 6 represent this phase. Let us see the growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of the organism can produce more cells in a variety of ways. The growth rate shown an increase that may be arithmetic or geometrical. Figure 15.1. Here is the diagrammatic representation of arithmetic growth, second is geometric growth, and third is stages during embryo development showing geometric and arithmetic phases. Arithmetic growth, only one cell divide. In geometric type of growth, all cells divide. Zygote divided. Geometric phase, all cells divide. Blue colored cells are capable of division, while the faint blue colored cells that lose the capacity to divide. In arithmetic growth, following mitotic cell division, only one daughter cell continued to divide while the other differentiate and mature. The simplest expression of arithmetic growth is exemplified by a root elongating at a constant rate. Look at figure 15.5 on plotting the length of the organ against time. A linear curve is obtained. Mathematically, it is expressed as L is equal to L0 plus RT. L is length at time t, L0 is length at time 0, R is growth rate, elongation per unit time. On x-axis, time is representative, while on y-axis, height of the plant. And the straight line graph is obtained. Let us now see what happens in geometrical growth. In most systems, the initial growth is slow, lag phase, and it increases rapidly, therefore, at an exponential rate, log or exponential phase. Here, both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of growth against time, we get a typical sigmoid or S curve. Figure 15.6. A sigmoid curve is a characteristic of living organism growing in natural environment. It is typical for all cells, tissue, and organs of a plant. Can you think of more similar examples? What kind of curve can you expect in a tree showing seasonal activities? Sigmoid curve like this. An idealized sigmoid growth curve typical of cell in culture and many higher plants and plant organs. On x-axis is time and on y-axis is representation of size weight of the organ, and S-shaped sigmoid shaped graph is obtained. Here is the lag phase, then exponential phase, and then stationary phase. The exponential growth can be expressed as W1 is equal to W0 ERT. W1 is final size, weight, height, number, etc. W0 is initial size at the beginning of the period. R is the growth rate, T is the time of growth, E is base of natural logarithmics. Here R is the relative growth rate and is also the measure of the ability of the plant to produce new plant material, referred to as the efficiency index. Hence, the final size of W1 depends on the initial size W0. Quantitative comparison between the growth of living system can also be made in two ways. First is measurement and the comparison of the total growth per unit time is called absolute growth rate. Absolute growth rate means measurement and the comparison of total growth per unit time. Total growth per unit time is called absolute growth, while the growth of the given system per unit time expressed on a common basis, example per unit initial parameter, is called a relative growth rate. Relative means per unit initial parameter. In figure 15.7, two leaves A and B are drawn that are of different sizes but show absolute increase in area in a given time. To give leaves A dash and B dash. However, one of them shows much higher relative growth rate. Which one and why? Relative growth rate is 5. Here also 5. Diagrammatic comparison of absolute and relative growth. Both the leaves A and B have increased their area by 5 cm in given time to produce A dash and B dash leaves. Here they have represented that. Conditions for growth. Why do you not try to write down what you think are necessary conditions for growth? This list may have water, oxygen, and nutrients as very essential element for growth. The plant cells grow in size by cell enlargement, which in turn requires water. The rigidity of the cells helps in extension growth. Thus, plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrient, macro and micro essential elements are required by plant for the synthesis of protoplasm and act as a source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain stages of growth. Let us see differentiation, dedifferentiation, and redifferentiation. The cells derived from root apical and shoot apical meristem and can be undifferentiated and mature to perform specific function. This act leading to maturation is termed as differentiation. During differentiation, cells undergo few to major structural changes both in their cell wall and protoplasm. For example, to form a tertiary element, the cell would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell wall to carry water to long distance, even under extreme tension. Try to correlate the various anatomical features you encounter in plants to the function they perform. Plants show another interesting phenomena. The living differentiated cells that by now have lost the capacity to divide can regain the capacity de-differentiation of division under certain conditions. This phenomenon is termed as de-differentiation. Regaining the capacity to divide is called a de-differentiation. For example, formation of meristem, intrafascicular cambium, and cork cambium forms fully differentiated parenchyma cells. While doing so, such meristem tissue are able to divide and produce cells that once again lose the capacity to divide but mature to perform specific function, that is, get re-differentiated. List some of the tissue in a woody dicotyledonous plant that are the product of redifferentiation. How would you describe a tumor? What would you call the parenchyma cells that are made up to divide under controlled laboratory conditions during plant tissue culture? Recall in section 15.11. We have mentioned that the growth in plant is open, that is, it can be indeterminate or determinate. Now we say that even differentiation in plant is open because cell tissue arising out of the same meristem have different structure at maturity. The final structure at maturity of a cell tissue is also determined by the location of the cell within. For example, cell positioned away from the root apical meristem differentiates as root cap cells, while those pushed to the periphery mature as epidermis. Can you add a few more examples of open differentiation correlating the position of the cell to its position in an organ? Let us see the development. Development is a term that includes all changes that an organism goes through during its life cycle, for germination of the seed to senescence. Diagrammatic representation of the sequence of processes which constitute the development of a cell of a higher plant is given in figure 15.8. It also applicable to tissue organs. Plants follow different pathways in response to environmental or phases of life to form different kinds of structure. This ability is called plasticity. Example, heterophyly in cotton, coriander, and larkspur are the example of plasticity, that they follow different responses due to environmental conditions. In such plant, the leaves of the juvenile plant are different in shape from those in mature plant. On the other hand, difference in shape of the leaves produced in air and those produced in water in buttercup also represent the heterophilous development due to environment. Figure 15.9 represents this phenomenon of heterophyly is an example of plasticity. Let us see the sequence of the developmental process in a plant. Here they have represented the meristematic cell, then plasmatic growth, differentiation, expansion, elongation, maturation, mature cell, senescence, death. In heterophyly in Langspan and buttercup is shown in the figure. This is the juvenile and adult. Terrestrial habitat, water habitat. According to environment, they have changed the shape of their leaves. Thus, growth, differentiation, and development are very closely related events in the life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plants, that is both growth and differentiation, is under the control of intrinsic and extrinsic factors. The former include both intracellular, genetic, or intercellular factors, chemicals such as plant growth regulators, while the latter include light, temperature, water, oxygen, nutrients, etc. Let us see plant growth regulators characteristic. The plant growth regulators (PGRs) are small simple molecules of diverse chemical composition. They could be indole compounds, indole 3-acetic acid (IAA), adenine derivative N6-furphylamine, purine, kinetin, derivative of carotenoids, abscisic acid ABA, terpenes, gibberellic acid GA3, or gases ethylene C2H4. Plant growth regulators are variously described as plant growth substances, plant hormone or phytohormone in literature. The plant growth regulators can be broadly divided into two groups based on their functions in a living plant body. One group of plant growth regulators are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting, and seed formation. These are also called plant growth promoter. Example: auxin, gibberellin, and cytokinin are plant growth promoter. The plant growth regulators of the other group play an important role in plant responses to wounds and stresses of the biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The plant growth regulators abscisic acid belong to this group. The gaseous plant growth regulators ethylene could fit either of these groups, but it is largely an inhibitor of the growth activities. So you have to remember that the ethylene is largely a plant growth inhibitor. The discovery of plant growth regulators. Interestingly, the discovery of each of the 5 major groups of plant growth regulators have been accidental. Also, this started with the observation of Charles Darwin and his son Francis Darwin, when they observed that the coleoptiles of canary grass responded to unilateral illumination by growing towards the light source, photoperiodism. After a series of experiments, it was concluded that the tip of the coleoptile was the site of transmeval influence that caused the bending of the entire coleoptile. Figure 15.10 represents this. Auxin was isolated by F. W. Wendt from tips of coleoptile of oat seedlings. Here they have represented experiment used to demonstrate that the tip of coleoptile is the source of auxin. Arrow indicates the direction of light. The growth occurred toward the direction of light. The Bacchanae foolish seedling disease of rice seedling was caused by the fungal pathogen Gibberella fungicoriae. E. Corsoa, 1926, reported the appearance of symptoms of the disease in rice seedling. When they were treated with sterile filtrate of the fungus, the active substance were later identified as gibberellic acid. F. Cook and his co-worker observed that from the internodal segment of tobacco stems, the callus, a mass of undifferentiated cells, proliferated only if, in addition to auxin, the nutrient medium was supplemented with one of the following extract of vascular tissue: yeast extract, coconut milk, or DNA. Miller et al. In 1955 later identified and crystallized the cytokinins promoting active substance that they termed the kinetin. During mid-1960s, 3 independent researchers reported the purification and chemical characterization of 3 different kinds of inhibitor. Inhibitor B, abscission; second, dorming. Later all the 3 were proved to be chemically identical. It was named abscisic acid ABA. H. Cousins in 1910 confirmed the release of a volatile substance from ripened oranges that hastened the ripening of stored unripened bananas. Later, this volatile substance was identified as ethylene, a gaseous plant growth regulator. Let us study some of the physiological effects of these 5 categories of plant growth regulators in the next section. Physiological effect of plant growth regulators. Auxin. Auxin, from Greek word auxin, to grow, was first isolated from human urine. So auxin is isolated from human urine. The term auxin is applied to the indole 3-acetic acid and to other natural and synthetic compounds having certain growth regulating properties. They are generally produced by the growing apices of the stem and roots, from where they migrate to the region of their action. Auxin, like IAA and indole butyric acid IBA, have been isolated from plants. Natural derivative of auxins are IAA, IBA, isolated from plants, while NAA, naphthalene acetic acid, and 2,4 dichlorophenoxyacetic or synthetic oxygen. You have to remember the example of natural oxygen and synthetic oxygen. All these oxygens have been used extensively in agriculture and horticultural practices. They help to initiate rooting in stem cutting, an application widely used for plant propagation. Oxygen promote flowering, example in pineapple. They help to prevent fruit and leaf drop at early stage but promote the abscission of older mature leaves and fruits. In most higher plants, the growing apical bud inhibits the growth of the lateral axillary bud, a phenomenon called apical dominance. Removal of shoot tips, decapitation, usually result in the growth of lateral buds, figure 15.11. It is widely applied in tea plantation. Hinge mark making, can you explain why? Because we need tea leaves, so we apply this. Apical dominance in plant. A plant with apical bud intact. Second, a plant with apical bud removed. Note the growth of lateral buds into branches after decapitation. Here they have represented the decapitation. Oxygen also induces parthenocarpy. Example, in tomato, they are widely used as herbaceous 2,4-D, widely used to cool dicotyledonous weeds, does not affect mature monocotyledonous plant. It is used to prepare weed-free lawns by gardeners. Oxygen also controls xylem differentiation and helps in cell division. Gibberellins. Gibberellins are another kind of promotory plant growth regulator. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They are denoted as GA1, GA2, GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellins to be discovered and remain the most intensively studied form. All gibberellin acid are acidic. They produce a wide range of physiological responses in the plant. Their ability to cause an increase in length of the axis is used to increase the length of grape stalks. Gibberellins cause fruit like apple to elongate and improve its shape. They also delay senescence, thus the fruit can be left on the tree longer so as to extend the market period. Gibberellic acid 3 is used to speed up the malting process in brewing industry. Sugarcane store carbohydrate as sugar in their stem. Spraying sugarcane trunk with gibberellins increase the length of the stem, thus increase the yield by as much as 20 tons per acre. Spraying juvenile conifer with gibberellic acid hastens the maturity period, thus leading to early seed production. Gibberellins also promote bolting, internode elongation, thus prior to flowering in beet, cabbage, and many plants with rosette habit. Cytokinins. Cytokinins have specific effect on cytokinesis and were discovered as kinetin, a modified form of adenine, a purine. From the autoclaved herring sperm DNA, kinetin does not occur naturally in plant. Search for natural substances with cytokinin-like activity lead to the isolation of zeatin from corn kernels and coconut milk. Since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in a region where rapid cell division occurs. For example, root apices, developing shoot apicals, young fruits, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and advantageous shoot formation. Cytokinin helps overcome the apical dominance. They promote nutrient mobilization, which helps in delay of leaf senescence. Ethylene. Ethylene is a simple gaseous plant growth regulator. It is synthesized in large amount by tissue undergoing senescence and ripening fruits. Influences of ethylene on plant include horizontal growth of seedlings, swelling of the axis, and the apical root formation in dicot seedling. Ethylene promotes senescence and abscission of plant organ, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruit. The rise in rate of respiration is called a respiratory climatic. Ethylene breaks seed and bud dormancy. Initiate germination in peanut seeds, sprouting of potato tuber. Ethylene promotes rapid internode petiole elongation in deep water rice plant. It helps leaves upper parts of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plant to increase their absorption surface. Ethylene is used to initiate flowering and synchronizing fruit set in pineapples. It also induces flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used plant growth regulator in agriculture. The most widely used compound as source of ethylene is ethiopion. Ethiopion is an aqueous solution, is readily absorbed and transmitted within the plant and release ethylene slowly. Ethiopian hasten fruit ripening in tomato and apple and accelerate abscission in flowers and fruits. Thinning of cotton, cherry, walnut. It promotes female flowers in cucumber, thereby increasing the yield. Abscisic acid. As mentioned earlier, abscisic acid ABA was discovered for its role in regulating abscission and dormancy. But like other plant growth regulator, it also has other wide-ranging effect on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. Abscisic acid inhibits seed germination. Abscisic acid stimulates the closure of stomata and increases the tolerance of the plant to various kinds of stresses. Therefore, it is also called the stress hormone. Abscisic acid plays an important role in seed development, maturation, and dormancy by inducing dormancy. ABA helps seeds to withstand desiccation and other factors unfavorable for growth. In most situations, abscisic acid acts as an antagonistic to gibberellic acid. We may summarize that for any and every phase of growth, differentiation, and development of plants, one or the other plant growth regulator has some role to play. Such roles could be complementary or antagonistic. These could be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where more than one plant growth regulator interact to effect that event. Example: dormancy in seed, buds, abscission, senescence, apical dominance, etc. Remember, the role of plant growth regulator is of only one kind of intrinsic control along with genomic control and extrinsic factor. They play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development by plant growth regulators. Some of such events could be vernalization, flowering, dormancy, seed germination, plant movement, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factor on initiation of flowering. Let us see the photoperiodism. It has been observed that some plants require a periodic exposure to light to induce flowering. It is also seen that such plants are able to measure the duration of exposure to light. For example, some plants require the exposure to light for a period exceeding a well-defined critical duration, while others must be exposed to light for a period less than this critical duration before the flowering is initiated in them. The former group of plants are called long day plant, while the later ones are termed short day plants. The critical duration is different for different plants. There are many plants, however, where there is no such correlation between exposure to light duration and induction of flowering response. Such plants are called day neutral plants. Figure 15.12 is shown that. It is now also known that not only the duration of the light period but that the duration of the dark period is also of equal importance. Hence, it can be said that flowering in certain plants depend not only on the combination of light and dark exposure but also their relative duration. This response of plant to period of day-night is termed as photoperiodism. It is also interesting to note that while shoot apices modify themselves into flowering apices prior to flowering, that is shoot apices of plant by themselves cannot precede photoperiods. The site of perception of light, dark duration are the leaves. It has been hypothesized that there is a hormonal substance that is responsible for flowering. The hormonal substance migrates from leaves to shoot apices for inducing flowering only when the plant are exposed to necessary inductive photoperiod. Let us see the figure. Here they have represented the flowering. Long day plant where flowering occur above the critical photoperiod. Below no flowering occur. In short day plant, above critical photoperiod no flowering, below flowering is occurred. While day neutral plant, no effect of critical photoperiod. Let us see the vernalization. There are plants for which flowering is either quantitatively or qualitatively dependent on exposure to low temperature. This phenomenon is termed as vernalization. It prevents precocious reproductive development late in the growing season and enable the plant to have sufficient time to reach maturity. Vernalization refers specially to the promotion of flowering by a period of low temperature. Some important food plants, wheat, barley, rye, have two kinds of varieties, winter and spring varieties. The spring variety are normally planted in the spring and come to flower and produce grain before the end of the growing season. Winter variety, however, if planted in spring, would normally fail to flower to produce mature grains within a span of a flowering season. Hence, they are planted in autumn. They germinate and over winter come out as small seedlings, resume growth in the spring, and are harvested usually around midsummer. Another example of vernalization is seen in biennial plants. Biennial are monocarpic plants that normally flower and die in second season. Sugar beet, cabbage, carrot are some of the common biennials. Subjecting the growing of the biennial plant to a cold treatment stimulate a subsequent photoperiodic flowering response. Let us see the seed dormancy. There are certain seeds which fail to germinate even when external conditions are favorable. Such seeds are understood to be undergoing a period of dormancy which is controlled not only by external environment but are under endogenous control or condition within the seed itself. Impermeable and hard seed coat. Presence of chemical inhibitors such as abscisic acid, phenolic acid, para-ascorbic acid, and immature embryos are some of the reasons which cause seed dormancy. This dormancy, however, can be overcome through natural means and various other man-made measures. For example, the seed coat barrier in some seeds can be broken by this absorption and caused by microbial action and passes through digestive tract of animal. Effect of inhibitory substance can be removed by subjecting the seeds to chilling condition or by application of certain chemicals like gibberellic acid and nitrate, changing the environmental conditions such as light and temperature or other method to overcome seed dormancy. Let us see the last part of this chapter summary. Growth is one of the most conspicuous events in any living organism. It is an irreversible increase expressed in parameters such as size, area, length, height, volume, cell number, etc. It conspicuously involves increased protoplasmic material. In plant, meristem are the site of growth. Root and shoot apical meristem, sometimes along with intercalary meristem, contribute to the elongation growth of the plant axis. Growth is indeterminate in higher plants. Following cell division in root and shoot apical meristematic cell, the growth could be arithmetic or geometrical. Growth may not be and generally is not sustained at a high rate throughout the life of cell, tissue, organ, organism. One can define three principal phases of growth: the lag, the log, and the senescent phase. When a cell loses the capacity to divide, it leads to differentiation. Differentiation results in development of structure that is commensurate with the function the cell finally has to perform. Generally, principle of de-differentiation for cells, tissue, and organs are similar. A differentiated cell may be de-differentiated and then redifferentiated. Since differentiation in plant is open, the development could also be flexible. That is, the development is sum of the growth and differentiation. Plant exhibit plasticity in development. Plant growth and development are under the control of both intrinsic and extrinsic factor. Intercellular intrinsic factor are the chemical substance called plant growth regulators (PGR). There are diverse groups of plant growth regulators in plant, principally belonging to 5 groups: auxin, gibberellins, cytokinins, abscisic acid, and ethylene. These plant growth regulators are synthesized in various parts of plant. They control different differentiation and developmental events. Any plant growth regulator has diverse physiological effect on plant. Diverse plant growth regulators also manifest similar effect. Plant growth regulators may act synergistically or antagonistically. Plant growth and development is also affected by light, temperature, nutrition, oxygen status, gravity, and such external factors. Flowering in some plants is induced only when exposed to certain duration of photoperiod. Depending on the nature of photoperiod requirement, the plants are called short day plant, long day plants, and day neutral plants. Certain plants also need to be exposed to low temperature so as to hasten flowering later in life. The treatment is called as vernalization. In this way, we completed the reading of plant growth and development chapter. So if you like the video and want more videos like this, so subscribe to Medical Dev. Thank you.